So yes, from plant kingdom in our previous classes, what did we deal? About systems of classification. What is the first system of classification, ma? Artificial system of classification, also addressed as sexual system of classification. Who are the proponents of them, ma? Aristotle and Linnaeus. Amongst them, who has proposed, who has taken androecium structure into consideration? Linnaeus. Whether it is bilobed, trilobed, monocolpate or bicolpate. Okay. What is the major drawback of artificial system of classification? Equal weightage to vegetative characters and reproductive or sexual characters. Next is what? After this we have learnt? Natural system of classification. Proponents? Bentham and Hooker. They have considered both internal and external characters into consideration. Third system is phylogenetic system of classification. Who are the proponents? Engler, Prandtl, Hutchinson, Tucktagen. These are the, you remember Engler, Prandtl enough. Okay. And then after they have considered what? Along with external and internal, they have also considered phylogenetic relationships into consideration. Okay. And after this we have learnt about taxonomy. Whenever the terms classification or taxonomy comes, there is no confusion at all. Classification ante entana? Grouping the living organisms based on the similarities and dissimilarities. What similarities? Based on morphological similarities, anatomical, embryological, all kinds of similarities and differences, we are grouping them and placing them into separate categories. That is classification. Okay. Arranging the group of living organisms into different ranks. It can be any rank. That rank can also be addressed as taxon. It can be from species to kingdom. That is known as taxonomy. Okay. In that, three types of taxonomy we have learnt for plant kingdom. What are they? Numerical taxonomy, cytotaxonomy, chemotaxonomy. Amongst these three, which is the advanced level? Numerical taxonomy. Also addressed as phonetics. In this, what is the major advantage? Hundreds of characters are considered at a time. Okay, because every character is assigned with a numerical or an alphabet, so all characters are considered at a single time. Okay. Second is what? Cytotaxonomy. What are the three criteria we will look in the cytotaxonomy? In simple terms, we can also say chromosome structure, number and behavior. DNA or chromosome, one and the same, structure, number, behavior. Okay. Then chemotaxonomy. In a single cell, all the cellular constituents will be there, be it the organelles, be it the chemical constituents. So, analyzing them and taking them into criteria, placing the organisms in different ranks is what? Chemotaxonomy. Clear with systems, no? Apart from this zone, you will not get extra questions, be it statement based or match the following. Okay. After this, we have learnt about 5 divisions of plant kingdom classified into cryptogams and phanerogams. Cryptogams 3. What are they, ma? Algae, Thallophyta, Bryophyta, Pteridophyta. Amongst these 3, which are non-vascular cryptogams? Bryophytes. Non-vascular terrestrial cryptogams are Bryophytes. Primitive plant species are algae. First vascular terrestrial cryptogams are Pteridophytes. These 3 groups are also addressed as non-spermatophytes because they are not seed producing. Okay. Then spermatophytes, phanerogams, flowering plants are 2 divisions. What are they, ma? Gymnosperms and Angiosperms. One more question. Tell me, what are the first embryophytes? First embryophytes. Bryophytes. Bryophytes are first embryophytes. First archegoniates. First archegoniates. Pteridophytes. First archegoniates are also bryophytes. First embryophytes, first archegoniates are what? Bryophytes. First vascular cryptogams are what? Pteridophytes. Here you see, ma, you should be clear on male and female gametophyte or of all the 5 divisions. Okay. Male gametophyte and female gametophytes. okay, of algae, bryophytes, pteridophytes, fourth, gymnosperms, angiosperms. It's okay. So, algae, male gametophyte is antherozoid. Female gametophyte is, if it is oogamous type of reproduction, oogonium is the female gametophyte. This is also addressed as sperm cell, okay. And here in bryophytes, what is the male gametophyte again? Antherozoid. Here also it is antherozoid, is the male gametophyte. Female gametophyte is archegonia, sorry, this is antheridium, antheridium. Antheridium, archegonium. Be clear with the terms, beta. Male gametophyte, female gametophyte. What does it mean? Male gametophyte is bearing the male gamete. Female gametophyte, gametophyte, phyte means plant body, gamete bearing, okay. Female gametophyte, female gamete bearing. What will be the female gamete? Egg cell will be the female gamete. Antherozoid or sperm cell will be the male gamete. In bryo and pterido, you can see jacketed sex organs enclosing this male and female gamete. Whereas in algae, do you see any such jacketed sex organs? No, except in advanced classes, like in Chara you see nucleus and globule, okay, but we are talking about the primitive forms, okay, they do not have the jacketed sex organs, fine. Here bryophytes and pteridophytes you have what? Antheridium and archegonium. So what are the first archegoniates, ma? Bryophytes are first archegoniates. First archegoniates means in which division did you see the first archegonium? Bryophytes. And what is the female gamete? Here antherozoid, here the male gamete is antherozoid, female gamete is egg cell. Got the difference between gamete and gametophyte, everyone? Here it is antherozoid again, here it is archegonium. In gymnosperms, male gametophyte is pollen grain. Inside pollen grain only, no, male gamete will be present. So gametophyte is pollen grain. And female gametophyte is archegonium here, again the same. Inside archegonium only you will have the egg cell. Angiosperms, if you see, which are the advanced classes, male gametophyte is pollen grain, female gametophyte is embryo sac. Inside embryo sac only you will have the female gamete, that is egg cell. If in specific you have to know, pollen grain is the male gametophyte. Inside which cell of pollen grain you will have male gametes? Generative cell. Pollen grain will have two cells, right beta? What are they? Vegetative cell and generative cell. Inside generative cell only it will replicate and it will form male gametes. Clear? You have understood this? If this picture is in your mind, you will easily understand the life cycles of bryo, pterido, gymno, angio. Done everyone? Did you take it, ma? Yeah. So now few questions from algae. Algae we have learned 3 classes. Okay, what are they? Chlorophyce, Pheophyce, Rhodophyce. What is the storage body of chlorophyce? Pyrenoids, they will be storing starch besides proteins besides starch. Pheophyce, cell wall. Cellulose, above which what hydrocolloid is there? Algin. Hydrocolloids of red algae. Agar, carrageen, funorine. Agar, carrageen and funorine. Next, what is the storage body in red algae? Floridian starch, which exhibits structural similarity with glycogen. It exhibits structural similarity with glycogen. Next, chlorophyce pigmentation. Chlorophyll A, B. Pheophyce pigmentation, C. Fucoxanthin is the major pigment which is giving which color to which color? Olive green to various shades of brown. Along with it, it also has carotenoids and xanthophylls. Clear till here, ma? Tell me the unicellular green algae. Unicellular green algae. Chlamydomonas chlorella. Okay. Isogamous type of reproduction with non-motile gametes in green algae. Spirogyra. Spirogyra. Okay. Isogamous motile gametes in green algae. Eulothrix. Oogamous in green algae examples. Fucus and volvox. Very good. And anisogamous examples? Eudorina. Okay. Then tell me what are the examples of brown algae? Ectocarpus, Dictyota, no, Polysiphonia is for red algae, Laminaria, Sargassum, Fucus. Amongst this, which exhibits diplontic life cycle? Fucus. Which has air bladders or nematocytes? Fucus. Which exhibits dichotomous branching? Dictyota. Okay. Which has all the 3, holdfast, type and frond? Laminaria. Tell me, what are the structures that are formed because of profusely branched ectocarpus? Kelps. Kelps. Kelps are the profusely branched ectocarpus which extend over 100 meters also. So this is about brown algae. Tell me amongst them which is rich in iodine? Kelps. Tell me what are the unicellular green algae that are used as protein supplements by space travelers? Chlorella and spirulina. Now, tell me which red algae are the sources of agar? Giladium, Gracilaria. Gracilaria and Giladium. Good. And tell me what is the advanced class in green algae? Charophyce, example is Chara. Advanced in red algae, Polysiphonia. Clear till here, ma? Everybody, revised algae. Now, you are okay with the male and female gametophytes, male and female gametes or of all the 5 divisions. Clear, right, everybody? Now, Bryophytes. See, I will give you ample time at the end. Focus only on the class. Focus only on the class. These are the primitive land plants, amphibians of plant kingdom. Why are they known as amphibians of plant kingdom? They are dependent on water for fertilization. The antherozoids, which are the male gametes, stored inside the antheridium, they are released into the water body. Antherozoids are mostly biflagellate. They will swim all across the water bodies and they will reach the egg cell. This fertilization process is known as zooidogamous oogamy. Zooidogamous oogamy. So, as it is dependent on water for fertilization, these are known as what beta? Amphibians of plant kingdom. Second is, these are archegoniate, embryophytic, atracheophytic. Remember, the first vascular plants are pteridophytes. What is the common vascular structure or bundle that it is having is tracheids. All the vascular plants will have tracheids. Pteridophytes, Gymnosperms, Angiosperms are tracheophytes. Bryophytes are atracheophytes because they don't have tracheids. So, these are archegoniate, embryophytic, atracheophytic cryptogams. You should also remember these are the first archegoniates and first embryophytes. Clear, no? Next, non-vascular terrestrial cryptogams, this you know, and habitat is mostly shaded, mostly in shaded areas. They grow in damp, humid, shaded localities. Habit, if you observe, you can see root-like, leaf-like, stem-like structures. Root-like structures are rhizoids, stem-like structures are phylloids, leaf-like structures are phylloids, stem-like structures are colloids. Clear with the intro part, ma? And you know two classes of bryophytes, those are liverworts and mosses. Okay? Mostly unicellular rhizoids you observe in liverworts. Multicellular rhizoids you observe in mosses. Clear, no? Bryophytes. Next, thallus you see, ma, thallus. It is dorsiventral. Markantia has what type of thallus? Dorsiventral thallus, which is closely appressed to the substratum. And this also shows dichotomous branching. Dichotomous branching. This is the dorsal part, this will be the ventral part. Rhizoids are present where, ma? In the dorsal part. And gymma cups are present where? In the dorsal part. Each gymma cup will have numerous gymma, many hundreds of gymma. Many gymma. One gymma will give rise to 2 thalli. One gymma will give rise to 2 thalli. Remember this. Each gymma cup will have numerous gymma. One gymma will give rise to 2 thalli. Clear with the thallus, ma? And porella, this example, whoever are writing EAPCET, you remember. NEET students not required. These are the leafy members. This also gives tiny leaf-like appendages. Tiny leaf-like phylloids are present, which are present on the stem-like structures. Over the colloids, phylloids are present. This is the structural description of porella. And Markantia is heterothallic. Heterothallic means both male reproductive structure, female reproductive structure are present on 2 different thallus. Okay. And here you remember rhizoids ploid is haploid because the complete thallus structure is haploid. Rhizoid structure is haploid, gymma cups haploid, antheridiophore haploid. Complete haplontic structure. Okay, gametophytic structure. Gametophyte is the main plant body. So all this will have what ploidyma? Haploid structures. It is heterothallic. Heterothallic, dioecious you can say, unisexual. All these are synonyms. Okay, heterothallic, dioecious, unisexual, these are what? Synonyms. All means one and the same. Heterothallic means both the different thalli have 2 different structures. Dioecious means the same. Unisexual also means the same. Clear with the structural description, everyone? Next. Here in Markantia, vegetative reproduction is by fragmentation and by gymma. In your NCRT, the structural description of gymma, of protonema, of prothallus are important. You will get statement based questions from these 3 areas. The description, statement description in the NCRT, those liners which are describing gymma, protonema which is the primary gametophyte of bryophytes, prothallus which is the primary gametophyte of pteridophytes, these 3 are important. How can you describe a gymma? Green multicellular asexual buds. These are green multicellular asexual buds. They develop on the small receptacles called as gymma cups. Mature gymma from gymma cups, they will detach from the parent body and germinate into new individual. Can you see 2 different thalli that are produced from a single gymma? Okay. Sexual reproduction is, so how can you describe gymma, ma? Green multicellular asexual buds. Okay, it is green multicellular asexual buds. Next is homothallic and heterothallic structures of liverworts. Of course, Markantia is what? Heterothallic or dioecious. Homothallic or monoecious include Riccia. It is dioecious or heterothallic. It has antheridiophores and archegoniophores. This is the antheridium. Inside antheridium what is present? Antherozoids are present. These antherozoids should be dispersed from antheridium, yes or no? How they will be dispersed? Via elaters. Elaters are the structures which will help in the easy dispersal of antherozoids into the water bodies. Okay, and this is the structure of antherozoid. Can you see the biflagellate condition? Yes, right. And next is archegoniophore. Inside archegoniophore you will have egg cell at the bottom. Can you see a single archegonial cell inside which the egg cell is present, and here this is the antherozoid. Okay. So once antherozoid reaches the egg cell, so where is the site of fusion? Inside archegonium. Site of fusion means where antherozoid is fusing with the egg cell. Egg cell is present here. Always male gamete will reach the female gamete. Okay. So this is via what? Zooidogamous oogamy, and site of fusion, gametic fusion, is inside the archegonium. Understood everybody? Any doubts anywhere? And post fusion, what is the first cell of sporophytic generation? Zygote. Zygote is the first cell of sporophytic generation, and zygote undergoes mitotic division, forms out embryo. This develops into a diploid sporophyte. Sporophyte is dependent on gametophyte. Understood everybody about Markantia? Listen carefully. You have antheridiophore, archegoniophore. Antheridiophore will be bearing what? Antheridium. Inside antheridium you have antherozoid. Complete ploid is haploid, that is gametophytic structure. It is bearing male gamete. About archegoniophore. Archegoniophore has archegonium, inside which egg cell is present. Right, ma? This is present inside the water bodies. This antherozoid will be reaching out via zooidogamous oogamy, and it will enter into the egg cell. Okay. Post fertilization, what is the first cell form? Zygote. Zygote is the first cell of what generation? Sporophytic generation. And where is the site of fusion? Inside the archegonium. Zygote undergoes mitotic division, forms out diploid embryo, then it matures to diploid sporophyte. Understood everyone? Any doubts anywhere? Yeah. Next, mosses. Liverworts are clear, no ma? Mosses you see. In mosses, which are the advanced category of bryophytes, you can see both the stages in gametophyte. First is primary gametophyte, addressed as juvenile stage. Primary gametophyte is addressed as juvenile phase, and then adult leafy stage is known as gametophore. Okay. This juvenile phase has what? Protonema. Protonema is described as creeping, green, branched, frequently filamentous stage. Remember the description, important for statement based. And this develops directly from what? Spores. Spores will directly develop into the protonema. Spore ploidy will be haploid. Protonema ploidy will be haploid. Gametophyte ploidy will be haploid. Got it? Leafy stage. This later develops to what? Leafy stage, which is upright slender axis bearing spirally arranged leaves. If I can take the example of funaria, this is the protonema, which is creeping green branched structure. This undergoes lateral budding, okay, and this develops into what? Leafy gametophyte. Ploidy of this is haploid. Leafy gametophyte is also haploid. This bears multicellular rhizoids. It has a main axis and it has leaf-like structures. Leaves are, how leaves are arranged here? Spirally arranged. Got it? Understood the primary structure and secondary structure, ma? This gametophore, which is the adult leafy stage, bears the sex organs which are antheridia and archegonia. Okay, it bears the sex organs antheridia and archegonia. Now you see, the multicellular or uniseriate filamentar hair-like structures present over the body of gametophyte are known as paraphyses. Nothing but the hair-like structures over the gametophyte are what? Paraphyses. Now you see, from the gametophyte what will be developed? Post fusion. Once antherozoid comes, fuses with the egg cell, from here zygote is formed, develops to embryo, embryo develops to sporophyte. Sporophyte ploidy is diploid. It has what structures? Foot, seta and capsule. Foot, seta, capsule. Foot ploidy is diploid, seta diploid, capsule diploid. Okay. Inside capsule, you will be having cells. These cells are addressed as spore mother cells. Okay. The spore mother cells inside the capsule itself, they will undergo what type of meiosis, ma? Sporic meiosis. They will undergo sporic meiosis and they will produce 4 haploid spores. They will produce 4 haploid spores. These haploid spores, landing onto the suitable substratum, they will develop into what? Protonema. From protonema, it develops to leafy gametophyte. Leafy gametophyte bears sex organs, antheridium and archegonia. Both are bearing what? Sterile jacket layers. Antheridium will bear what? Antherozoid. It goes to the egg cell, fuses, forms zygote, embryo. Embryo develops to sporophyte. Sporophyte is dependent on gametophyte. In Funaria, if you see, sporophyte has 3 structures: food, seta and capsule. Inside capsule you have what? Spore mother cell, which undergoes what meiosis? Sporic meiosis. If you remember in algae, what type of meiosis takes place? Zygotic meiosis. Sporic meiosis undergoes the reductional division and 4 haploid spores are formed. In liverworts, for the dispersal of antherozoids, you have elaters. Here you have peristomic teeth. For the dispersal of spores, what do you have? Peristomic teeth. Peristomic teeth help in spore dispersal. Got it? Understood the life cycle, my everybody? Okay. This is about mosses. These synonyms you have to remember. Funaria is also known as cod moss, sphagnum, peat moss, bog moss, cotton moss. Polytrichum is addressed as hair cap moss. Polytrichum is hair cap moss, of which sphagnum is bisexual. It has both male reproductive structures, female reproductive structures over the same plant body. Okay. Sphagnum is what? Bisexual or monoecious. Funaria and Polytrichum are dioecious or unisexual. Clear everybody with liverworts and mosses? Next is economic importance of bryophytes. From sphagnum itself, if you consider the previous year questions, you can easily see 2 to 3 questions on peat moss, bog moss, cotton moss. Okay. So, mosses play major role in plant succession. You know, lichens are the pioneers of plant succession, be it primary or secondary succession. Pioneers of plant succession are what? Lichens. They will start the life. After lichens, the complete bed is formed by what? Mosses. Okay, mosses form a green structure, complete green structure, which are creeping over the earth bed during the succession time. So, after lichens, it is mosses which play major role during plant succession. That is one role. So, as it is promoting plant succession, it will prevent the soil erosion. And about sphagnum, it is addressed as peat moss because it is used as good manure and fuel. It is also known as bog moss because it builds soil in rock creases and fills ponds and lakes. It is known as cotton moss because it is a good adsorbent of water. You can use sphagnum for transshipment of commercially important plants like tulips and orchids. Okay. Dry sphagnum is used for transshipment of seedlings and cut plants, mainly for commercially important plants. These days, for all the celebrations, mostly if you have so much of money, you can invest on commercially important plants, some exotic plant species like tulips, orchids, like that. So, this will be used for that. If anybody are having nurseries and all, they will be using this. Next. So, if you completely visualize the life cycle of bryophytes, listen carefully everyone, you will get a complete picture here. We will start from primary gametophytic structure, which is creeping, branched, filamentous structure. This is what ma? Protonema. Buds on protonema, can you see? It forms lateral budding and develops into what? Gametophore. Gametophore, which is the leafy gametophytic structure. Okay. You can see two jacketed sex organs here, antheridia and archegonium. Sperm cell will reach as the egg cell by what? Zooidogamous oogamy. Zooidogamous oogamy. Post fertilization, what is formed? Zygote is formed. This zygote matures to embryo. What is the first cell of sporophytic generation? Zygote, which is diploid. And this develops to embryo. Embryo develops into sporophyte. Ploidy of this is diploid. Inside the capsule of sporophyte, what are present? Spore mother cell undergoes reductional division, forming 4 haploid spores. These spores are released via elaters in liverworts, via peristomic teeth in mosses. Okay, and these spores will develop into what? Protonema like structures. So, can you see there are equal stages of gametophyte and sporophyte? In the complete life cycle, you can see equal stages of gametophyte and sporophyte. Okay, so that is why what is the life cycle ma? Haplodiplontic life cycles. Clear till here ma? Bryophytes, everybody are clear with it now? Now, we will move on to pteridophytes. Any doubts anywhere? I will give you time to copy. I will give you time to take the notes. If you have understood, you say yes. We will move on. Next is pteridophytes. How can you describe pteridophytes ma? These are the first terrestrial plants to possess vascular tissues. In simple terms, first terrestrial vascular cryptogams. Embryophytic, archegoniate, tracheophytic. Bryophytes are atracheophytic. These are tracheophytic. Seedless vascular plants. Habitat, you see, cool, damp, shady places, and they are also present in sandy soil-like structures. Main plant body or dominant phase is sporophyte, and gametophyte is independent. Dominant phase is sporophyte. In bryophytes, what is the dominant phase? Gametophyte is dominant phase, because their sporophyte is dependent on gametophyte. Here if you observe, both sporophyte and gametophyte are independent to each other. Both are what? Independent to each other. And most of the phase you can see what? Sporophytic phase only. So, what is the dominant phase? Sporophyte. It is independent of gametophyte. Gametophyte is also independent of sporophyte. This includes what type of examples? Club mosses, horsetails, ferns. Main plant body is sporophyte, and this sporophyte is differentiated to root, stem, leaves. Here root, stem, leaf ploidy will be diploid, because it is the sporophytic plant body. Where are the spores present? Over the leaves. Over the leaves. That is why here leaves are called as what? Sporophylls. Leaves are addressed as sporophylls. They can be small structures or large leaves. Small leaves or large leaves. Small leaves are known as microphylls. Large leaves are known as macrophylls. Microphylls example, Selaginella. Macrophylls, ferns. Roots are what? Adventitious. Understood everyone? Next. Leaves and leaflets. Leaves and leaflets. They have sporangia. What are those structures known as ma? Sporophylls. Development of sporangia occurs where? Over the sporophylls. The development can be leptosporangiate or eusporangiate. If a single cell is involved in the development of sporangium, it is known as what? Leptosporangiate. If group of cells are involved in that, it is known as eusporangiate. Once, to make you clear, in pteridophytes, what is the main plant body? Louder. Sporophyte is the main plant body. It is differentiated to what structures? Root, stem, leaf. Over leaves what are present? Sporangia are present. Sporangia are present. That is why leaves are known as what? Sporophylls. If they are small, you are calling it as microphylls. Large, they are known as macrophylls. Till here to clear, right? Yeah. How sporangia is developed over the leaf structure? Based on that, you have two things. What are they? Eusporangiate and leptosporangiate. Eusporangiate, if group of cells are involved in sporangia formation, it is known as eusporangiate. Leptosporangiate, from a single cell only sporangium will be developed. Understood ma? Eusporangiate and leptosporangiate. Next, over the sporophyll you have sporangia. What are sporangia? Diploid cells, and each cell in the sporangia has many spore mother cells ma. It has many spore mother cells. These sporangia are attached to what? Leaf structures. Okay. One of the spore mother cell we will take. Its ploid is what? Diploid. It undergoes what meiosis? Sporic meiosis again. Sporic meiosis. Forms what? Haploid spores. How many? 4 different types of spores are formed. Okay. First heterosporous condition observed in Pteridophytes only. Till Bryophytes all are homosporous. The spores which are released are same. They are alike. But here you can see first heterosporous condition means it is releasing small spores and large spores. Small spores are known as microspores, large spores megaspores. Microspores will develop into what? When they fall on the substratum, microspores fall on the substratum. Okay, they will be developed to prothallus, then to primary gametophyte. What gametophyte? Antheridium or male gametophyte. Microspores develop to what ma? Male gametophyte. Megaspores develop to female gametophyte. Understood? First differentiation you have observed in Pteridophytes. Okay. So to remember the examples easily, Selaginella and Salvinia are the heterosporous condition. They are first observed in Pteridophytes. Okay. These are the examples in the NCERT. But along with this, 2 more examples. For that the mnemonic I give is MAS. Okay. Marsilia, Azolla, Selaginella, Salvinia. Marsilia, Azolla, Selaginella, Salvinia. Clear, no? So you are clear with homosporous, heterosporous conditions, sporophyll, leptosporangiate, eusporangiate, everything is clear, right? Now, homosporous, in homosporous, gametophyte is what type ma? Monoecious. Monoecious means what? Bisexual or homothallic. Okay. Heterosporous are dioecious because 2 separate you are getting, right? Male gametophyte and female gametophyte. This is also known as what? Unisexual. Clear, no? Next. Over the equisetum structures. Equisetum. These are hair cap moss like structures. Strobili are cones. In this, sporophylls are into distinct compact structures. If you see Selaginella and other pteridophytes, over microphylls and macrophylls, you can see sporangia structures. If these sporangia are into compact cone-like structures present above the plant, that is known as what? Strobili or cones. This is an advanced evolutionary feature connecting to what? Gymnosperms. In Gymnosperms also you can see strobili and cones. Examples include what ma? Selaginella and equisetum. Clear till here? Next. So spores landing onto the substratum develop into prothallus. How do you address prothallus? How do you describe it? Small, inconspicuous, multicellular, free-living, photosynthetic gametophyte structure, thalloid gametophyte. Once everybody, small, inconspicuous, multicellular, free-living, photosynthetic, thalloid gametophyte. Okay. Sex organs are multicellular, jacketed, sessile sex organs. Heterospores means megaspores develop into female, microspores develop into male gametophyte. Here you see what type of oogamy? Zooidogamous oogamy. And when zygote develops to embryo, the site of fertilization is zygote only. The site of fertilization is archegonia only, sorry. Inside archegonia only fertilization takes place. And the first cell of sporophytic generation is zygote. Zygote develops to embryo. This story is clear to us. Once embryo is formed in bryophytes, that embryo will directly develop into sporophyte. Embryo is not staying inside the archegonia. But here in pteridophytes, embryo stays for certain variable period of time. For some period, embryo is staying inside the archegonia. Okay? But that will not sustain for longer time because female gametophyte doesn't have any integuments to protect the embryo. Does it have any integuments? No. Whereas if you see ovule in the angiosperms, it is integument. It has integuments, okay, to protect the embryo sac. But here there are no integuments. That is why embryo sustains only for certain period of time. And then embryo develops into what? Sporophyte. Clear till here? Zooidogamous oogamy. Then, so this phenomena where embryo is staying inside the archegonia for variable period of time is addressed as what? Precursor to seed habit. Here seed is not formed, but a habit is shown. A habit of seed formation is shown. That is addressed as what? Precursor to seed habit. So in pteridophytes, these two major things you have to remember, ma. First heterosporous condition and precursor to seed habit. Many a times the questions from pteridophytes are asked from these two regions. Okay, and examples of pteridophytes. Ploidy is important. So this sporophyte is what structure? Multicellular, well differentiated dominant phase. What are the four classes of pteridophytes? Louder. Louder. Coelopsida. Coelopsida example. Lycopsida. Spinopsida. Pteropsida. Okay, no, till pteridophytes. Next about gymnosperms. Once for your clarity, a quick repetition on pteridophytes. Sit straight everyone. Pteridophytes' dominant plant body is sporophyte. From there we will start. In sporophyte, spore mother cells, where are they present? Over sporangia. They are present over leaf-like structures, sporangia. Sporangia, based on that development of sporangium, it has two types. What are they? Eusporangiate, leptosporangiate. If group of cells involved in sporangium formation, one cell involved in that sporangium undergoes what division? Sporic meiosis. Sporic meiosis forms haploid spores. Haploid spores, they are released either in homosporous condition or heterosporous condition. If they are released in heterosporous condition, it can be either microspores or megaspores. Microspores develops into male gametophyte. Megaspores develop into female gametophyte. Right, ma? Till here it is clear. Once these spores landed on the substratum, spore ploidy is diploid. They are landing and developing into a primary gametophyte, that is prothallus. Prothallus develops into mature gametophyte. Mature gametophyte can again develop into antheridia, archegonia, the cycle continues. Got it? So what type of life cycle it has? Haplodiplontic life cycle. Two major features you have to remember is what? Heterosporous condition, precursor to seed habit. Okay, embryo is not sustaining for longer time because absence of integuments. Okay, clear now everyone? Remember all the examples: heterosporous conditions, leptosporangiate, eusporangiate. This is the concept of pteridophytes. You have to remember. And synonyms also, synonyms of bryo and pteridophytes. Now gymnosperms. These are also embryophytic, tracheophytic, archegoniate, phanerogams. Phanerogams are spermatophytes. Spermatophyte means what? Phyte means plant body, sperma means seed. Seed bearing plant body. Okay. So these are phanerogams or spermatophytes. You can observe primitive seed formation here. Okay. Why seeds are not covered with ovary wall? Seeds doesn't have ovary wall. So before fertilization, ovules are not enclosed by ovary walls. Inside archegonia, you have ovule-like structures. These ovules are not enclosed by ovary walls. So seed is not enclosed by ovary wall. And post fertilization, that is the reason ovule develops to seed, right? Ovary wall is not there, so the seed coat or fruit wall is absent post fertilization. You have to remember one thing, ovule develops to seed. There is no ovary wall, so there is no seed coat. Before fertilization, there is no ovary wall. Clear, no? Plants are of medium sized or tall trees or shrubs. You should remember what is the smallest gymnosperm and largest gymnosperm. Smallest angiosperm and largest angiosperm. Smallest gymnosperm is Zamia. Largest gymnosperm is Sequoia. Living fossil is Ginkgo. Ginkgo is the living fossil. Ginkgo biloba. Okay. This is the introductory part of gymnosperms. Next, root you see, you have taproot system only. Pinus has mycorrhizal association, means it has what? Fungal associations. Why fungal associations? If you see, this is the taproot. If fungal associations are over the peripheral structures like this is known as ectomycorrhiza. Okay. If the fungal strands are going into the cells, this is known as endomycorrhiza. Endomycorrhiza, one example I can give you, vesicular arbuscular mycorrhiza, VAM. Endomycorrhiza example. Okay, because they will be dwelling into the vascular strands. What is the purpose of this symbiotic association? The fungi which is associated will extend the surface area of the roots, of the lateral roots. It will extend the surface area of the roots. Thereby, it will increase the mineral absorption. It will increase the water absorption. What indirectly the fungi is getting? Shelter over the roots. This is the mycorrhizal association observed in what, ma? Pinus. What is the main purpose? To increase the absorption of water and minerals. Got it, ma? Next, coralloid roots. Okay, what are they associated with? They are associated with blue-green algae or cyanobacteria. What is the purpose of this association? Nitrogen fixation. Coralloid roots observed in cycas. Clear about the root system. As they are associated with fungi or blue-green algae, what are absent? There are no root hairs or root caps. No root hairs or root caps. Next is stem. You can see unbranched stem in cycas, branched stem in Pinus and Cedrus. Next, stems have U-stele, which is advanced stele. Stem has U-stele, which is advanced stele. Vascular bundles are conjoint. Conjoint means both xylem and phloem are at the same radius. Collateral, they are present side by side to each other like this. Xylem, phloem. Xylem, phloem. This is collateral. They are present side by side to each other to conduct what? Lateral conduction. Conjoint means xylem, phloem at the same radius. Collateral, xylem, phloem side by side to each other. Vascular bundles are side to each other to conduct lateral conduction. Open form of vascular bundles means in between xylem and phloem you have what? Cambium, vascular cambium. Conjoint, collateral, and open type of vascular bundles. In xylem, what are absent? Vessels are absent. Many a times asked question. Okay. Tracheids, xylem fibers, xylem parenchyma are present, but vessels are absent. In phloem, what are absent, ma? Companion cells are absent. Instead of companion cells, what are present? Albuminous cells are present. Sieve tubes are absent. Instead of sieve tube, what is present? Sieve cells. In angiosperms, you can see sieve tubes, companion cells, phloem fibers, phloem parenchyma. But here only what are present? Sieve cells and albuminous cells. Secondary growth occurs in both stem and root because it has open type of vascular bundles. Understood about the anatomical description of gymnosperms? What kind of stele it has? How are vascular bundles? In xylem and phloem both are complex tissues because it has 4 tissues in each. Amongst which in xylem what are absent? Vessels. In phloem what are absent? Companion cells. And sieve tubes. Instead of companion cells what are present? Albuminous cells. Instead of sieve tube cells what are present? Sieve cells. Sieve cells. Okay. Does secondary growth occur in gymnosperms? Yes. Because of cambium. Leaves you see, ma. Next. Leaves are pinnately compound leaves in cycas. Needle-like leaves you can see in conifers. Fan-shaped leaves are present in ginkgo. Why in conifers you can see needle-shaped leaves? This will reduce the surface area of leaves, thereby minimizing the transpiration process. Okay? And then leaves are adapted to withstand the extreme temperature, humidity, wind. Thick cuticle, sunken stomata is present. Thick cuticle and sunken stomata helps to reduce water loss. Okay. Clear? This is about leaves. Next. Only heterosporous condition is observed in gymnosperms. Only heterosporous condition. In pteridophytes you can see homosporous, heterosporous. Only heterosporous in gymnosperms. Microspores develop into male gametophyte, megaspores develop into female gametophyte. Here you can see sporophylls are of compact structures. All the spores are into compact structures called as strobili or cones. They are arranged in spirally along, they are arranged around spiral axis which are known as strobili or cones. This is a very important concept. So only heterosporous condition is observed in gymnosperms. How microsporophylls and megasporophylls are present you see. They are arranged in compact cone-like structures, strobili we have seen. No. If it is male gametophyte, these are compactly arranged to and they are addressed as what? Male strobili. Male strobili is also known as microsporangiate or male cone. This consists only male gametes. Okay? If the strobili is consisting only male gametophytes, only pollen grains, you will call it as what? Male strobili, microsporangiate, male cones. They are present inside microsporophylls. Microsporophylls compactly arranged to male strobili, microsporangiate, male cone. In microsporangiate, all the cells which are present are diploid. Microsporophylls are diploid, right? So microsporangia are diploid. Amongst that, one cell I am taking, microspore mother cell. Undergoes what meiosis again? Sporic meiosis. Forms microspores. These microspores are haploid. This develops into male gametophyte or pollen grains. Okay? Pollen grains are wing shaped in pinus. Okay? Pollen grains are what shaped in pinus? Wing shaped. This pollen grains will reach the archegonia. Before that, you listen to the structural description of megasporophyll also. Megasporophylls are compactly arranged to female strobili, also known as megasporangiate, also known as seed cone. If you consider this as the female cone or seed cone, inside female cone you can see the pore-like structure above, which is micropyle end, and it has integuments. Can you see integuments of this cone? Integuments are present, and each cone bears 2 to 3 archegonia. Inside female cone, all the cells which are present are known as nucellus. One of the nucellus will be differentiated into megaspore mother cell. It undergoes what? Sporic meiosis. Forms what? 4 megaspores, of which how many megaspores are functional, ma? 1 megaspore is functional. Okay. Once for your understanding, once again I will repeat. See, microsporophylls, sit straight. Microsporophylls compactly arranged to male strobili, microsporangiate, male cone. All the cells present inside that are known as microsporangia. One cell, microspore mother cell, undergoing sporic meiosis, forms haploid microspores. They will develop into haploid pollen grains. Okay, this is the male gametophyte. And here female reproductive structures. Female strobili, megasporangiate, seed cone, inside which you have all the nucellus cells, 2 to 3 archegonia, integuments are present, and pore-like structure above this is known as micropyle. This nucellus, one of the nucellus cell is differentiated to megaspore mother cell, undergoes sporic meiosis, forms megaspores, of which one cell is functional megaspore. Okay. Functional megaspore develops into what? Female gametophyte. This again bears 2 to 3 archegonia. So remember here, the functional megaspore, whichever is undergoing the division, okay, this will divide mitosis. This will undergo mitosis. It will undergo divisions. As this is haploid, this forms haploid cells. Okay, what type of cells it is forming? Develops into a female gametophyte. That structure, female gametophyte, it is bearing what? 2 to 3 archegonia, and the cells present here are known as what? Endosperm cells. Can you observe here, endosperm is forming prior to the fertilization. Endosperm is forming where? Prior to the fertilization, whose ploid is what? Haploid. So megaspore gives endosperm, functional megaspore giving rise to what? Endosperms. And inside this archegonial cells, what are present? Egg cell is present, and the pollen grain reaches the egg cell. This phenomena of fertilization is known as siphonogamous oogamy. Siphonogamous previously in Bryophytes and Pteridophytes, what type of oogamy it is? Zooidogamous oogamy. Their male gametes are motile. Here male gamete is non-motile. It is dispersed mostly via abiotic factors, mostly via wind. Okay, and they will reach this through this tube. That is why this is known as what? Siphonogamous oogamy. Clear till here, ma? Everybody? Yeah. So this is the life cycle. Listen carefully. Gymnosperms life cycle is diplontic life cycle. Mature sporophytes, ploid is what? Diploid. All are heterosporous. You can see male separate, female separate branches. Pollen cone and ovulate cone, also known as female cone. This has microsporangium, undergoes meiosis, forms microspores. Those microspores develop into pollen grains. You can see wing shaped pollen which is seen in Pinus. Okay. Pollen tube is formed, okay, at the time of fertilization to reach the archegonia. Now you see ovulate cone ploidy, diploid, undergoes meiosis. Inside this ovulate cone you have nucellus cell. One of the nucellus cell differentiates to megaspore mother cell, and that megaspore mother cell undergoes meiosis, forms megaspores, of which only one functional megaspore develops into female gametophyte. This has what? Archegonia inside it. Okay. Inside archegonia, egg cell is present. Via pollen tube, it is entering into the egg cell. Here also the site of fertilization is archegonium. First sporophytic stage is zygote. So if you can see, only this male gametophyte structure and female gametophyte structure are haploid phases. Rest all phases are diploid. Here after zygote is formed, zygote develops to embryo, embryo develops to seed, seed develops to mature sporophyte. Got it everyone? Understood, no? So except this haploid pollen grain male gamete, haploid megaspore female gamete, all the other phases are what? Diploid phases. So what is the life cycle? Diplontic life cycle. Clear about gymnosperms everyone? Then angiosperms. Listen carefully. Angiosperms you will also learn again in sexual reproduction of flowering plants. If you are learning here clearly, there it gets easy. These are embryophytic, non-archegoniate. Archegonia is not the female gametophyte here. What is the female gametophyte? Embryo sac. This is vascular, fruit bearing, phanerogams or spermatophytes. Smallest angiosperm is Wolffia. Smallest gymnosperm is Zamia. Very good. Largest angiosperm is Eucalyptus. Smallest angiosperm is Sequoia. Sequoia. Okay. Vascular bundles are complex tissues. Xylem has 4 tissues: xylem fibers, tracheids, vessels, xylem parenchyma. Phloem has phloem fibers, sieve tubes, companion cells, phloem parenchyma. Sieve tubes are enucleated. How the function of sieve tubes will be done? Via companion cells. That we learn clearly in anatomy again. What is the reproductive organ here? Flower. There male cone, female cone you can see, but here what is the reproductive organ? Flower is the reproductive organ. Where you can see the bisexual condition. Okay. Flower has how many whorls? 4 whorls, 2 non-essential, 2 essential. Non-essential are calyx, corolla. Essential whorls include andrecium and gynoecium. Andrecium structures are what? Stamen. It can be one stamen or many stamens. Each stamen will have what? Filament and anther. Same case. Pistil consists of carpels. Carpels. It can be monocarpellary or multicarpellary. Each carpel will have ovary, style, stigma. How double fertilization takes place? Just a second. I will directly explain you via life cycle. Everybody can see this? Yes? Yes or no?
Yes, ma'am.
See, anther will have what, ma? Pollen grains. Mostly pollen grains are dispersed in what cell stage? 2-celled stage. Vegetative cell and generative cell. How pollen grains are formed? We will learn there in microsporogenesis of sexual reproduction. Here, yeah, here only it is given. inside the anther, roughly I will tell you, listen, anther, you know 4 wall layers of anther? Okay. What are the 4 layers? Epidermis, endothelium, middle layers and tapetum. Tapetum is the innermost layer. Inside the tapetum you will have 4 sporogenous tissues. Ploidy of sporogenous tissue is sporogenous tissue diploid. They have what type of cells inside the sporogenous tissue? Microspore mother cells whose ploidy is diploid. Okay. This undergoes meiosis, forms what? Microspore tetrad. 4 microspore cells are formed, no? Haploid microspores. This microspores will again undergo one mitosis, forming tube cell, which is vegetative cell and generative cell. Okay. Generative cell again divides into 2 cells. Once it is forming a pollen tube and growing inside. Inside the pollen tube, generative cell again divides into 2 male gametes. 2 male gametes. Out of these 2 male gametes, if you see inside the ovule, mostly what type of ovule you observe, ma? What type of ovule you see? Inverted ovule in the angiosperms. Okay, inverted ovule. This is the micropylar end, this is the chalazial end. These are integument regions, and inside which all the nucellus cells are present, right? One of the nucellus cell differentiate to megaspore mother cell. Same story. Megaspore mother cell undergoes meiosis, forms 4 megaspores, of which 1 megaspore is functional. That 1 megaspore will undergo 3 sequential mitotic divisions, forms embryo sac inside the ovule. Okay. This embryo sac is how many celled condition? How many celled condition embryo sac? 7 celled, 8 nucleated condition. 3 antipodals and 3 egg apparatus, you will call it as 1 egg cell, 2 synergies. At the center, polar nuclei are present, which is known as central cell. 3 plus 3 plus 1, total 7 cells. Polar nuclei, 2 nuclei are present, so 8 nucleated condition. Okay. So this egg cell, 1 male gamete fuses with egg cell, 1 male gamete fuses with polar nuclei, which is addressed as triple fusion or double fertilization. After which, with the fusion of male gamete and egg cell, what is formed? Zygote is formed, whose ploidy is diploid. With the fusion of male gamete and polar nuclei, what is formed? Endosperm is formed, whose ploidy is triploid. Okay. Zygote develops to embryo, embryo matures to seed. Seed ploidy is diploid. Seed, when undergoes seed germination, develops into sporophyte. Sporophyte ploidy again diploid. Develops to mature sporophyte. Over sporophyte you can see flowers, diploid. So except the haploid microspores, pollen grains, haploid embryo sac and the megaspore, functional megaspore and haploid embryo sac, rest all phases are diploid. So the life cycle is diplontic life cycle. Understood everyone about angiosperm life cycle? Clear, no? Angiosperms are again classified into 2 classes. What are they? Dicots and monocots. Dicots will have what type of leaf venation you see? Reticulate venation. How many whorls are present? 4 or 5 whorls. Like how many whorls, how many structures are present over the flower? 4 or 5. Sorry, 3 or multiples of 3. We are talking about dicots, no? Dicots though it is 4 or 5, and monocots though it is 3 or multiples of 3, like poaceae members. Okay. Venation is parallel, and how many cotyledons are present? Single cotyledon. So this is the alternation of generations we are talking about. Gametophytic generation and sporophytic generation. Simple question for you people. Okay. Male gametophytes belongs to what generation? Gametophytic generation. Pollen grain belongs to? Pollen grain. Microspore. Gametophyte. Microspore mother cell Sporophytic generation. Megaspore mother cell. Capsule. Sporophytic generation. Okay. Female gametophyte. Female gametophyte. Gametophytic generation. I hope you understand. Protonema belongs to which generation? Gametophytic generation. Prothallus belongs to gametophytic generation. Okay. So, all the structures that you encounter in this chapter, you should be clear to which generation it is belonging to. When you will be clear? When you clear on the life cycles of all divisions. Okay. And when it comes to life cycles, you have understood the haplontic life cycle we have learned yesterday with algae. Exceptions are important. Zygotic meiosis, haplontic life cycle observed in all the algae. Examples you can take colonial algae, volvox, Spirogyra filamentous algae, species of Chlamydomonas unicellular green algae. Diplontic life cycle, Bryophytes, Pteridophytes, some algae like Ectocarpus, Polysyphonia, Kelps, Dictyota. Next, diplontic life cycle observed in Gymnosperms Angiosperms. Fucus is diploid, which is brown algae. Clear, right? Clear till here everywhere? This ends the chapter what? Plant kingdom. What you have to focus here more on the ploidies. You will get the calculation based questions by asking you about meiosis, calculations based on the meiosis and ploidies. Second is all the diagrams you have to be clear. What structures belongs to sporophytic generations, what structures belongs to gametophytic generations. Examples are very important of algae, pryo, pterido, gymno. Okay. Last but not the least, life cycles. These are the concepts that you have to focus from plant kingdom. Understood everyone? Tomorrow we will be starting with morphology. Till then you revise completely, okay, plant kingdom, and if you are having any doubts from the previous chapters of cell biology, plant physiology, living world also, biological classification and PK, you can get it to me, okay. 1 minute.